Hi everyone, we're going to, oh goodness, chapter 11. Dive into chapter 11. I'm going to switch into, I forgot what chapter we were doing. Switch into screen share mode right now. This PowerPoint is obnoxiously long, so I'm probably going to switch and stop the PowerPoint at some point and make it into at least two videos because the PowerPoint, as you see, is 228 slides long. I don't even know if I have the attention span to sit here and talk for that long. So we'll probably end up splitting it up a little bit. But this chapter is all about the nervous system and the divisions of the nervous system. First, there are anatomical divisions, the central and peripheral. The central nervous system is our brain and spinal cord. This is the majority of the integrating and processing of and coordinating of all of the sensory input and the motor output. And then the periphery is everything that's outside of the central nervous system. So I always forget to click. There we go. Let's talk about the subdivisions of the peripheral nervous system. So the peripheral nervous system can be divided into the sensory division and the motor division. The sensory division is bringing the information to the central nervous system. This can be through somatic sensory receptors, special sensory receptors, and visceral sensory receptors. The motor division can be divided into two categories, the somatic and the autonomic. The somatic is dealing with our muscles. The autonomic is all of the smooth muscles, cardiac muscle, glands, adipose, etc. So let's talk about some of the general functions. We have receptors, which are detecting the changes in the environment, internal or external environment, and that information is then sent to the central nervous system by the sensory division of the peripheral nervous system. Then there's some type of information processing that goes on. This is where your brain or spinal cord interprets that information and determines where that information needs to go. Either it needs to go up to our consciousness awareness or it needs to go to other places. Then the motor commands are carried out by the peripheral nervous system, and then the effectors are what's responding to those motor commands, like a muscle flexing or like a gland secreting, etc. Here you see the whole breakdown. You have some type of receptor, like a visceral receptor, a special sense receptor, or somatic receptor, communicating with the sensory division of our peripheral nervous system, taking up to the central nervous system for some type of information processing, and then responding with some type of motor command, either through the somatic or skeletal muscles or through the autonomic nervous system, which is our smooth muscle, glands, heart, etc. Whole process right there, just in a couple slides. All right, you can pause the video here, go back and review. I know I talk fast, but that's the joy of videos. You can go back and re-listen to it, right? Let's talk about the neurons, the kind of meat of our central nervous system. There are very specialized cells, and they're specialized for communication. There are three general regions to a neuron. You have the dendrites. The dendrites are receiving the information either from the environment or from another neuron. Then you have the cell body, which is where the nucleus is and all the important organelles. And then the axon is carrying the information away from the cell body, communicating that to other cells. The components of the neuron also include those dendrites. The dendrites are very highly specific. They're branched and they're receiving the information from all over the place. And when I say they're specific, I mean they can be shaped differently depending on which type of neuron we're talking about. We'll get to that in a second. The cell body includes something called the cytoplasm, and in muscle language, we call that the sarcoplasm. In neuron language, we're calling it the perikaryon, which translates to around the nucleus. So the organelles provide energy, obviously. They also synthesize and create the neurotransmitters. And there's an extensive cytoskeleton full of neurofilaments and neurofibrils, which are going to allow for the transport of information and materials down the axon. Let's talk about the axon. The axon begins at the axon hillock. It's the enlarged portion of the kind of proximal portion of the axon. It's right next to the cell body. Then the initial segment, which is where the first action potential is initiated. The axolemma is the specialized plasma membrane of the axon, and the axoplasm is the specialized cytoplasm of the axon. We also have telodendria, which are extensions at the end of our axon. These telodendria form axon terminals or synaptic terminals. There you see them, the telodendria on the distal end. If we look at the proximal end of our neuron, you see lots of dendrites. Dendrites fuse into the cell membrane where we have all of our organelles. Then we have that axon hillock, initial segment, all the way down our axon to the telodendria, axon terminals. Synapse is the gap between our neuron and the postsynaptic cell, which could be another neuron, or it could be like a muscle or some type of effector. So let's talk about how things get down that axon. It's requiring the use of neurotubules, which are the microtubules of the neuron. It's a process called axoplasmic transport, and it occurs both directions, up and down the axon. And that's, again, important for getting the neurotransmitters to the end of the axon. The synapse is the specialized site for communication between the neuron and some other cell. You have the presynaptic cell, which is the cell before the synapse. That is the one that's releasing the neurotransmitters. It has a presynaptic membrane where those neurotransmitters are released. And the synaptic cleft is the gap between the two cells. The postsynaptic cell is the cell after the synaptic cleft. This is going to have receptors for the neurotransmitters. Here you see where all the magic happens, the axon, axon terminal. Axon terminal generally has some mitochondria there for the active transport or exocytosis of these vesicles, which contain some type of neurotransmitter like acetylcholine, epinephrine, norepinephrine. The synaptic cleft, which is just that little gap between pre- and postsynaptic cells. And then our postsynaptic membrane, which will have specialized receptors for those neurotransmitters. Let's talk about the types of synapses. There are three main types of synapses. Neurons can synapse with another neuron. They can synapse with between a neuron and a muscle, or there can be a synapse between a neuron and a gland, so neuroglandular synapses. Collateral branches of axons will allow a single neutron to communicate with several other cells. So in other words, it's not a one-to-one ratio. There are typically more postsynaptic cells than there are neurons. Here you see what I mean. Here's our neuron, and it's synapsing with a collateral maybe to another, either another neuron or another effector in some way, shape, or form. Same thing with neuromuscular. Neuroglandular secretions usually don't have collaterals, but they could. So let's talk about how neurons are replaced. Typically, they're not replaced. They cannot divide. Most central nervous system neurons are incapable of mitosis. So if they're damaged or if they're injured in some way, they're gone. You're not going to get them back. There are some neural stem cells, but they're usually inactive. And so they just kind of sit there. Our sense of smell, the epithelium for our sense of smell, those do have stem cells, and they can be replaced because our sense of smell is such a hugely important sense in our survival. Our retinal cells can be replaced, although it's, again, very slow and efficient process. And parts of our brain involved in memory can also undergo mitosis and renew themselves, the hippocampus involved in memory. You can pause the video here, go back and review. Let's talk about how our neurons are classified. They're classified based on their structure. There's 4 anatomical classifications of neurons. You have anaxonic neurons. That prefix "an" usually means without. So these are small neurons that lack distinguishing features. So you really can't tell that they have axons or dendrites. They kind of look like a normal cell almost. Their functions are not very well understood. They are within the brain and within some of our special sense organs. There are bipolar neurons, which have 2 distinct processes. They have a dendrite process and an axon process. They're very rare, but they are in special sense organs. Then you have unipolar neurons. These have dendrite and axons that are continuous, so the cell body's lying off to one side. That initial segment is where the dendrites converge, and then the rest of the process is considered an axon. It looks kind of funny. You'll see the picture. Most sensory neurons are unipolar, and the axons can be very, very long, up to 1 meter, 3 feet in length. That's pretty long. And they mostly carry sensory information from like the tips of the toes all the way up to the spinal cord. So again, they can be pretty long. Multipolar neurons, that's like the one that I just showed you in that picture a few slides ago. That's where you have the typical dendrites, a cell body, and 1 axon. They usually have multiple dendrites, but just 1 axon, as I said. And this is the majority of our neurons. They can be very long as well. Here they are. Anaxonic, again, not very recognizable as a nerve cell. Then you have our unipolar, bipolar, multipolar. That's weird that slide isn't, it's not even labeled. Sorry about that. All right, let's talk about let's talk about functions of our neurons. They can really come in 3 different functions. They can be sensory, they can be motor, or they can be what's called an interneuron, which is relaying between the sensory and the motor. The majority of our neurons are interneurons. They're involved in that processing process. They're involved in that kind of analytical, logical, figuring out what to do in response to the sensory information or to the motor pathway. So let's talk about our sensory receptors. There's several different types of sensory receptors. There can be interoreceptors. These are monitoring various parts of the body inside. They're providing sensation about stretching, deep pressure, and pain within the body. Proprioceptors are about maintaining and monitoring our body's position. Are we sitting? Are we standing? Is our arm bent? Is our back sitting up straight, or are you kind of slumping in your chair like I am? Right? All of those things are proprioception, position of joints, muscles, etc. Exteroreceptors are monitoring our external environment. When I mentioned sensory receptors, that's probably what you thought of. You thought of hot, cold, right? Those are all exteroreceptors. So you have afferent nerve fibers. These are axons that are carrying sensory information into the central nervous system. They are arriving, afferent arriving, all right? A lot of students like to think afferent away. Afferent is arriving at the central nervous system. So it's the opposite of what you're probably thinking. There is a ganglion, which is a collection of cell bodies outside of the central nervous system within the periphery. The sensory ganglia contains cell bodies of those unipolar sensory neurons, somatic and visceral sensory motor neurons, which are very abundant. Then you have the interneurons. Interneurons are usually they're always within the central nervous system, so they're within the brain and spinal cord. They're, again, the relay between the sensory and the motor neurons, and they receive information from the periphery and relay it to the central nervous system, and they're responsible for those higher functions like memory and learning and emotions and all of those things. Motor neurons are where the cell bodies are located inside the central nervous system, as opposed to sensory, where they're outside of the central nervous system. The axons can go into a peripheral nerve, and that nerve bundle, a nerve is a bundle of axons in the peripheral nervous system. So again, we tend to think of the word nerve and we just think of, all the nervous tissue in general, but a nerve is specifically a bundle of axons in the peripheral nervous system. And you have somatic motor neurons, which are innervating our skeletal muscles under conscious control. You can consciously, command your body to do the robot or whatever. You also have visceral motor neurons. These visceral motor neurons are going into all the effectors other than our skeletal muscles. This is our glands, this is our heart, this is our digestive system, the adipose tissue, anything else that needs some type of input is all being done through this visceral motor control. They're carried through efferent fibers. Efferent are exiting the central nervous system. So you have afferent arriving, efferent exiting, and all of the afferent is sensory, all of the efferent is motor. Here is the whole pathway drawn out for you. So you have these sensory receptors. They can be intero, proprio, or extero. Some type of sensations going on. It's going to communicate to the sensory ganglia, which are within the peripheral nervous system. Remember, a ganglia is a cluster of cell bodies. That's going to transport its information to an interneuron, transfer the information to either the somatic motor neurons or the visceral motor neurons, and then those motor neurons are going to respond to the peripheral effectors. This is the whole process drawn out for you. You can pause the video here, go back and answer these 3 questions. Let's talk about the other type of nervous tissue. We have our nerve cells, or our neurons, but we also have these neuroglial cells, or glial cells, as they're sometimes called. These are the cells that support and protect the central nervous system. They protect the neurons, they nourish the neurons, they help make sure the neurons are well nourished and have what they need. They're abundant in both the central nervous system and the peripheral nervous system, and they're very diverse. And they make up about half of all the nervous tissue. So just as many of those as we have of neurons. There are 4 main types of neuroglial cells in the central nervous system. Ependymal cells. Ependymal cells form this kind of columnar epithelium called the ependyma. It's basically lining the fluid-filled passway where the cerebrospinal fluid is going to circulate. And it's the role of these ependymal cells, again, picture like an epithelial lining. That's just specialized for the central nervous system. The role of this is basically to monitor and adjust and really create and produce the cerebrospinal fluid. So there's 3 main types of ependymal cells. We have the ependymocytes, which are very long and slender. They have cilia to help circulate the CSF. Then we have tanycytes. These are non-ciliated. They're really only in one ventricle, and they just help to transport stuff between the cerebrospinal fluid and the brain. And then we also have specialized CSF producing ependymal cells. So these are special cells that produce the cerebrospinal fluid. Then we have microglia. Microglia are similar to monocytes and macrophages, so they have like an immunological function. They migrate into the central nervous system as it's being formed, and they persist as phagocytic cells. So they are basically little Pac-Man floating around our CSF, trying to gobble up any debris, anything that doesn't belong there, waste products, pathogens, etc. We also have astrocytes. Astrocytes are those types of cells that maintain the blood-brain barrier. The blood-brain barrier is ultimately the isolating barrier between the central nervous system, or the CSF, and the blood. So there's no connection. That's a lie. There's a few connections between the blood and the brain, but for the most part, when you cut open a brain, it's not going to be bloody, right, like the rest of our body. And that's because it's surrounded by that cerebrospinal fluid. And we'll talk about why and how and all that stuff later. But these astrocytes are the cells that help maintain that boundary. They basically provide structural support for the neural tissue. They regulate ion and nutrient and waste product levels, and they absorb and recycle neurotransmitters. They also help to form the scar tissue if the central nervous system has been injured. Oligodendrocytes are the last remaining neuroglial cell in the central nervous system. They provide a framework by stabilizing our axons. They also produce something called myelin. Myelin is basically a little, I think of it as bubble wrap going around the outside of our axons, protecting, insulating them, and speeding up the propagation of action potentials, which we'll get to later. I create something called a myelin sheath. So that myelin sheath is incomplete. It has little spaces in between it. So here's a myelin sheath. Here's my axon. Here's a myelin sheath. Here's a myelin sheath. So only a part of that axon is exposed. And the part that's exposed is called the node. The parts of the myelin sheath that is hiding that axon are called the internodes. Axons that have a myelin sheath are called myelinated. They appear white because of that fatty myelinated oligodendrocytes, and areas with lots of myelinated axons make up what we call the white matter of the central nervous system. Those oligodendrocytes may be absent from some axons, and those would be unmyelinated axons, and they appear gray. That's the gray matter. So here you see all of those neuroglial cells in action. See a multipolar neuron here surrounded by those oligodendrocytes. Oligodendrocytes, as you see, they can wrap around several different axons. So there are some membranes kind of off to the side, and they can kind of sprawl out and wrap around a couple different axons. You can see some astrocytes. I know without even looking at the label, those are astrocytes, because astrocytes look like little stars. They're kind of like little spacey star-looking cells. And then you have the ependymal cells with their cilia off to the side. Is that the only? Oh, the microglial cells. These are these little invaders. They kind of stick around and gobble up anything that's floating through. You can pause the video here, go back and answer these 3 questions. Let's keep going. Let's talk about the neuroglial cells of the peripheral nervous system. Instead of having oligodendrocytes, they have Schwann cells. It's the same thing, just in the peripheral nervous system. They cover the myelinated axons. They also participate in axon repair, which is minimal, but they can be repaired briefly by these Schwann cells. There's also satellite cells that surround the peripheral cell bodies. They help to regulate the environment around the neuron, kind of like the astrocytes. So let's talk about the difference between myelinated and unmyelinated axons. Myelinated axons are basically our speedy communicators of our peripheral nervous system. The myelin sheath is formed by Schwann cells in the peripheral nervous system and oligodendrocytes in the central nervous system. The outer surface of a Schwann cell is surrounded by something called a neurolemma. It's a connective tissue covering. So one single Schwann cell can wrap around a single internode, unlike the oligodendrocytes, which were kind of spread out and wrapping around several different axons. Unmyelinated axons in the peripheral nervous system will basically not be as chemically isolated, and they'll propagate their action potentials much slower. A single Schwann cell can enclose multiple segments of these unmyelinated axons. They help to stabilize the position of axons, and as I mentioned, they help to isolate the chemical boundary of the surrounding interstitial fluid. Here you see the myelinated axon and unmyelinated nodes. These are Schwann cells. Again, notice they just wrap around individually, and that outer covering is the neurolemma. Neurolemma is kind of like the cell membrane or plasma membrane of the Schwann cell. Think about it like that. So here you see the Schwann cell nucleus, the axon, and then the neurolemma is just that outer covering. Notice how this cell is wrapped and wrapped and wrapped and wrapped and wrapped around that axon. So it doesn't just wrap one time, it wraps several times. There it is again. Unmyelinated. These unmyelinated axons, again, they have the Schwann cells, but the axons themselves are not individually wrapped. So the Schwann cells are still there, but they don't have the same role in propagation of action potentials and protecting the axons like in the myelinated ones. So that's a big difference. Axon injury and repair in the peripheral nervous system is minimal, but the axon and the myelin can degenerate distal to the injury, so that will help increase blood flow to the area and increase the repair process. The Schwann cells will help proliferate around the original path of that axon, so they kind of fill in like a little shadowing, I guess, of where that axon used to be. Macrophages come in and gobble up all the extra debris, and then the axons can regrow in the path of that Schwann cell. So and then the Schwann cells will continue to wrap around that axon, and it may restore function or it may not restore function. So the repair process does not restore full function, is called Wallerian degeneration. It's just the type of degeneration that occurs if that neuron cannot repair itself, which is usually what happens. Here you see the whole process of Wallerian degeneration. Well, this is repair. So you have something happen to the nerve here, and so we have our Schwann cell kind of elongating and filling in that kind of shadowing effect, and the axon can continue to stretch out into that area, as you see here, and our Schwann cell wraps around it and protects it as it grows. There's the whole process again. Axon injury and repair is very limited regeneration properties in the central nervous system. So there's many more axons involved in the central nervous system. The astrocytes will produce a scar tissue, which can prevent the regrowth of the axons, and they basically are releasing chemicals as well to prevent the regrowth. So those astrocytes are a blocking mechanism to the regrowth of central nervous system axons and neurons. And you might say, well, why would you want that to happen? Well, it really is a protective measurement against tumor formation. It's okay if your brain's missing one or two neurons. It's not okay if your neuron is trying to regrow itself and mutates and turns into a tumor. So it's a repair mechanism or a defense mechanism. You can pause the video here, go back and review these 3 questions. We're moving into neurophysiology, so here's some learning outcomes for you. Let's talk about that membrane potential. We briefly got introduced to it in the muscular system. We were talking about the charge distribution of ions over the cell membrane and how there's more sodium outside the cell, more potassium in the cell, etc. And that unequal charge distribution creates what we call a membrane potential or a transmembrane potential. So that is just a review. This slide right here is just a review of what I just said. Inside the membrane, slightly negative. Outside the membrane, slightly positive. And that's due to the unequal distribution of sodium and potassium ions. Proteins and chloride have a little role, but not really. So the membrane potential and transmembrane potential is characteristic of all living cells. It, again, results from the differing compositions of extracellular and intracellular fluid, and there's active transport mechanisms to help hold this charge in place. Review: plasma membrane, outside is positive, inside's more negative. That's it. So let's talk about the role of the membrane potential and neuronal activity. Basically, changes in that membrane potential can trigger a muscle contraction, gland secretion, or transfer of information. So we have this idea of a resting membrane potential, or RMP, or resting potential. This is when a cell is undisturbed, its natural distribution of charges across the membrane. It varies from cell to cell. Typically, muscle cells and nerve cells have the same generalized resting membrane potential. All neural activities begin with a change in that membrane potential. Something happens that causes a change in ion concentrations across that membrane. We create what's called a graded potential. A graded potential is temporary, localized, and it may or may not cause an action potential. If that graded potential is big enough to hit what we call the threshold, it will then cause an action potential. But if it's not big enough, then it will just kind of go away. It's caused by just a small stimulus. If that stimulus, if you're very far away, if the potential is very far away from the stimulus, it's not obviously going to be as much of a change or as much of a graded potential as something that was very close to the stimulus. It's kind of like an echo effect. So the action potential will only be triggered if the graded potential reaches threshold or if it's big enough to change the polarity of the nerve cell itself. So the action potential is technically an electrical event that involves one location on that axon membrane. And then it spreads like dominoes down the axon to the axon terminals. At the synapse, the synaptic activity is typically involving the release of a neurotransmitter. So once that action potential gets down to the end of the axon, those neurotransmitters are going to be released, typically like acetylcholine, epinephrine, norepinephrine, etc. Then those neurotransmitters will bind to the postsynaptic cell, causing some membrane change in that cell and some type of response, either propagation of the action potential, contraction, etc. Or it could inhibit that cell. These action potentials could be inhibitory or excitatory, as we'll see. So the neuron basically has to do some degree of information processing. That's usually done by the interneuron when that information is relayed from the sensory input up into the central nervous system to be converted into motor output. But your neurons still individually have to do some degree of information processing. They have to integrate the stimuli in order to say, okay, this is what the cell's telling me to do. This is what this action potential is telling me to do. It's telling me to communicate this, or it's telling me to communicate this way to this muscle cell, or it's telling me not to do anything. So here you see this whole effect. Some information comes in, maybe it's some type of stimulus, some type of gated channel, etc., creating a graded potential, a small change. If that small change is big enough, loud enough, etc., and that axon hillock / initial segment, the action potential will propagate down the axon, triggering the release of neurotransmitters at the synaptic cleft, and that postsynaptic cell will undergo some information processing where it either says, I'm being inhibited, or I'm being told to do something, or whatever. It will respond in some way. You can pause the video here and answer these 3 questions. So let's get into detail about this resting membrane potential. Resting membrane potential is really based on, as I said, sodium and potassium, and to a small degree, proteins and chloride. So extracellular fluid has very high concentrations of sodium and chloride. Cytosol inside the cell has high concentrations of potassium and proteins. Proteins and chloride are negatively charged. The sodium and potassium are positively charged. But sodium and potassium are the main factors involved here. Proteins and ions can't move freely across the cell membrane. So there's a charge gradient and some pumps involved in maintaining that difference. So our resting potential is a form of potential energy, kind of like water holding, or a dam holding back water. It's measured in millivolts. Our body's cells range in resting membrane potential from -5 volts to -100 volts. The resting potential of muscle cells and nerve cells is around -70 millivolts. So maintaining that potential involves very passive forces like diffusing sodium and potassium through leak channels, which are just open and allowing the free flow of sodium and potassium. And there's also an active process which involves the sodium-potassium exchange pump. Those leak channels are the major reason why the membrane potential exists, because passive channels are open in the plasma membrane. They're always open. Their shape, structure, and size determine which ions can pass through them. Potassium ions diffuse out of the cell, again, because it's passive. We have a cell that has very high concentration of potassium. Outside is high concentration of sodium. So if those leak channels are open, they're going to change position naturally. Here you see what's happening. So enter the sodium-potassium exchange pump. What the exchange pump does is it pumps those ions against their concentration gradient. We have lots of sodium outside the cell, lots of potassium inside the cell. They're wanting to leak like this, but the pump is forcing them back to where they belong, forcing them back on the correct side of the membrane, forcing the sodium out, forcing the potassium in. And it's a ratio of 3 to 2. 3 sodium ions for every 2 potassium ions. So in other words, 3 positive charges are leaving and only 2 positive charges are coming in, which means our cell's slightly negative. Here you see the whole thing in action. We have these leak channels here. Again, notice this, the sodium is leaking in passively. Potassium's leaking out passively. So what does the sodium-potassium exchange pump do? It says, hey, get back here. 2 of your potassium need to come back in. 3 of your sodium need to get out. And so overall, at the rates of diffusion, our cells maintain at around negative 70 millivolts. This is a video that you can watch on masteringEMP.com. So that electrochemical gradient, as I said, is a form of potential energy. It's determined by those ions, concentrations inside and out of the cell. There's a chemical side, which is just the concentration of ions, and then the electrical side, which is the charge distribution. So it's electrochemical. It's both. That equilibrium potential is the membrane potential at which the ions' electrical and chemical gradients are equal and opposite. So this would be where there's no net movement. When the ion's contribution is no longer entering or leaving the cell. Equilibrium for potassium is around negative 90 millivolts. Equilibrium for sodium is around positive 66 millivolts. Keep those numbers in your mind. It's going to play a role. So our potassium ion gradient is at normal resting potential. Small electrical gradient is directed into the cell. Larger chemical gradient is directed outside of the cell. So there's a net chemical gradient outside of the cell. That large chemical gradient is pushing out. If the membrane were freely permeable to potassium, potassium would continue to leave the cell until that membrane potential meets negative 90 millivolts. That's what equilibrium potential means. It would just keep leaving until it reaches negative 90. Same thing with, and here you see just the breakdown of those leak channels. Again, potassium would just continue to leave until we reach negative 90, and then it would be equal in and out, equilibrium. Sodium ions at normal resting potential, electrical gradient is directing them into the cell, right? Because they have a high concentration out of the cell. The chemical gradient is directed into the cell. So there's a strong net movement of sodium into the cell for equilibrium forces. So if the membrane were freely permeable to sodium, and that sodium-potassium pump wasn't working, the sodium would continue to enter the cell until the membrane potential reached positive 66 millivolts. So again, here we're looking at these leak channels just being open. That sodium is just freely coming in. It won't reach equilibrium until that cell is at positive 66. I don't know why they made those a couple slides. It's kind of silly. You can pause the video here, go back and answer these 3 questions. So we'll talk about gated channels. So far we've talked about 2 ways that sodium and potassium get into and out of the cell. One is the leak channel, open, passively. They're always flowing like that. Sodium's always coming in, potassium's always going out, and that exchange pump's always pumping them in the opposite direction. Then we also have gated channels. Gated channels are usually closed during a resting potential. They can be opened or closed in response to a stimulus. If there's a change in ion movement or if there's a change in membrane potential, they can be opened or closed. There are 3 different types of gated channels. There are chemically gated channels, voltage gated channels, and mechanically gated channels. So chemically gated channels are also called ligand gated channels. These are open when special chemicals bind to them. So an example would be a receptor that binds acetylcholine at the neuromuscular junction. Acetylcholine binds, that's a chemical, and then our sodium channels open on that postsynaptic cell, on that muscle cell. Remember, the sodium rushes in and that causes the calcium to be released, etc., etc. These are most abundant on the dendrites and cell body of a neuron, these chemically gated channels. It's the site of most synaptic communication. Here you see what I mean. This is our sodium channel. Acetylcholine binds, it opens and allows that sodium to come in, or whatever ion. I was just using sodium as an example here. Oops, wrong way. Sorry about that. All right, then we have voltage-gated channels. Voltage-gated channels are characteristic of excitable membranes. These are membranes that are capable of generating or spreading an action potential. They open and close in response to the overall membrane potential or charge. These are most important for neurons. They're voltage-gated potassium, voltage-gated calcium, voltage-gated sodium channels. They have two gates that function independently. There's activation and inactivation gates. So here you see these are voltage-gated channels where at 70 millivolts, our gate's closed. We go to 60 millivolts, our gate's open, freely permeable, totally open, and then at plus 30 millivolts, our channel is now inactivated, not letting any more in or out. So mechanically gated channels are open in response to physical distortion. So this would provide sensory information like touch, pressure, vibration. So these are mostly our receptors that are on our hands and our skin, right? The channel's closed, but then as soon as there's pressure applied, there's touch, there's vibration, there's something that's going to distort the membrane, and mechanically gated channels are going to open, and as soon as that pressure is removed, the channels will close. The distribution of gated channels along the neuron varies. The chemically gated channels are typically found on the cell body and on the dendrites, which makes sense. The dendrites are going to receive the neurotransmitters from a presynaptic cell, and then they'll respond accordingly. The voltage-gated channels of sodium and potassium are found along the axon, which makes sense so that action potential can propagate. And then the voltage-gated calcium channels are found at the axon terminals because calcium is needed in order for acetylcholine or whatever neurotransmitter to be released. We kind of left that step out of the process when we were looking at neuromuscular junctions in our muscle chapter, but we're going to learn more nuts and bolts in this chapter. Obviously, it's the whole focus of the chapter. So here you see the distribution of channels. Chemically gated, voltage-gated, voltage-gated. Again, the neurons that have mechanically gated are going to be our sensory neurons that are mostly associated with our skin, and some of our visceral sensory receptors in our viscera, like our stomach and intestines. You can pause the video here, go back and answer these questions. Let's talk about graded potentials. A graded potential is also called a local potential. It changes in membrane potential. It's a change in membrane potential that doesn't spread far from the site of the stimulus. So it's just kind of a little spark, right? Nothing's ignited yet. So graded potential can be produced by like an opening of a sodium gated channel, or a gated sodium channel, sorry. So these graded potentials can be produced by those chemically gated sodium channels. At rest, the gated sodium channels are closed. Then when the membrane is exposed to some type of chemical, they open, and the sodium ions enter the cell, brings in a rush of positive ions, positive charge, and that's going to overall reduce the membrane potential from -70, bringing it to a higher number, a number closer to 0. That process is called depolarization. So once the sodium ions in the cell spread out away from the channels, the sodium moves through the channels, and this is basically going to produce what's called a local current, where the sodium's moving through the channels. More extracellular sodium can move in along the outer membrane towards the cell, replacing the ions that have entered the cell. It's just going to continuously flow into the cell. It's called a local current, all this flow of positive charges. So here you see what I mean, this graded potential. We have lots of sodium outside the cell. Then we have these voltage-gated channels here. Our potential changes just a little bit, opening those sodium channels. That's going to change the membrane potential here. It's going to open these sodium channels, so on and so forth. The whole thing is going to propagate down the axon. And now we have this kind of flow of sodium into our cell and down our cell and down our axon, producing this local current. Graded potentials can also be produced by chemically gated sodium channels. So the degree of depolarization is going to decrease with distance away from the stimulus. As ions enter at one location, they spread out in all directions. That's obviously going to dilute the signal. So the impact on membrane potential is proportional to the size of the stimulus. The larger the stimulus, the more channels are going to open. The more sodium enters, the greater the depolarization, etc. So here you see maybe 2 neurons coming in and causing that excitation or depolarization at that axon hillock. So when the membrane changes in membrane potential, it could open the chemically gated sodium channels, which leads to membrane depolarization. When that chemical stimulus is removed, the excess sodium is transported back out of the cell, and then we get more and more negative, closer to resting membrane potential, which we call repolarization. So the chemically gated potassium channels actually stay open a little bit too long and let too much potassium out, and we call that hyperpolarization. So here's the whole process. You get some type of stimulus. That stimulus is going to cause sodium channels to open, so sodium rushes into the cell. Since sodium's positive, it's going to increase our overall potential, increase our charge on our cell, and the stimulus is removed. So the sodium ions are transported back out of the cytosol. We get back to resting membrane potential, and then maybe a chemical stimulus opens potassium channels. When the potassium channels are open, we get hyperpolarization, and the stimulus is removed, so the potassium moves, leaves the cell, and we get back to membrane potential, resting membrane potential. Here's the whole process graphed out for you, step by step. I drank a lot of coffee this morning, I'm having chest pains. Happens all the time. If I die in the middle of the video, hopefully I don't. You'll probably know. Let's talk graded potential. Here's the chart that comes from your book, and it's just giving you the characteristics of a graded potential. Now, remember, a graded potential is localized, small, it doesn't propagate. So it's just kind of, again, it's like a little spark that doesn't ignite the entire fire. Just a little bit of a change. You can pause the video here, go back and review. I think I'm going to stop the video here. Don't be scared about me talking about chest pains. It happens all the time. I'm totally fine. But we're about halfway through the presentation, so this is a good stopping point. Oops, didn't want to do that. I'll just do this. This is a good stopping point for this PowerPoint, so it's not too long of a lecture. So our next little mini lecture from chapter 11 will start with action potentials as opposed to graded potentials. So, all right. I will see you in a little bit.